Okay, good. Yes, so many thanks for the invitation. It's great to be here and participate in this symposium. So let me start that actually it's one of the key features of the French community is really to go towards electron microscopy at various levels. And so the initiative to install Untitled Chapter at CSRF is very welcome, and it's part actually of a larger initiative in France, I would say. So you have the infrastructure for integrated structural biology, Frisbee. It's over 5 centers, so Grenoble and Strasbourg are the 2 centers that are also European centers, which include various tools across various methods, as you can see. And a key event has been 2 days ago, the adoption of the Instruct ERIC status, which is a major event for all of us and for the whole of Europe, actually, for structural biology. So in terms of electron microscopy in France, we have set up a roadmap to enhance instrumentation. And so there are 3 levels of instrumentation that we suggest. And this is a plan in terms of project. It's called France Cry EM. It's a national project focused and very strategic for our field to install high electron microscopes at national facilities, to enhance support for screening microscopes and for direct electron detectors in various places. All right. And so with that general aspect of infrastructure, which is very important, and I think it's the right place to speak about that, I'd like to move to the work we have been doing in my group over the last couple of years and present you a high-resolution structure of the ATS ribosome. This is something that started a long time ago. So we work actually on 2 aspects, on transcription and on translation complexes. And today I focus entirely on the aspects of ribosome complexes, which are really key to understand if you want to understand mechanism of action of antibiotics, of chemical reactivity, specificity of ligand interactions, function and dysfunction. So the human system is really a complex system, and for a long time it was thought hardly feasible. And it's really electron microscopy that made the difference to get there. Why it was so difficult is because of these long RNA expansion segments here, which basically create a lot of disorder in your structure. Finally, after a lot of sample optimization, we have been able to even crystallize it at low resolution, but then moved on with electron microscopy. And this is actually the first Titan Chapter in France, was installed in 2013 and allowed us to straight away collect data on these optimized samples, which gave an average structure, average resolution of 3.6 angstrom with detailed maps, as you can see here, with side chains, which for us was a major event to get there ever. With refinement of the atomic model of all 80 proteins and RNAs, as you can see here. And some of the highlights of that analysis is actually the interaction here of the intersub unit, for example, between helices, between the 40S and the 60S subunit, where you have involvements of 2' OH positions on the RNA, which is an interesting feature. So it's not magnesium ion mediated, basically. You can also look at conformational changes here between proteins that will transfer side chain interactions once you swap between 2 positions, 2 conformations, pre- and post-translocation states. There's also a very interesting feature as the E-side TNA that was contained in this complex that actually interacts here through base pairing and stacking interactions, including this typical interaction with the tyrosine. So I'm mentioning that because it's a key place for antibiotic interactions, and you will see that in a second. So we tried then to make a feasibility study. Could we look at ligand complexes in the human ribosome? And so we did that with the first classical ligand, which is used in lots of translation assays, cycloheximide. And by a lot of sorting, we managed to get a subcomplex which had the ligand bound and in which the TNA was released. So basically, ligand binding will induce release. So here you have the 2 complexes at full resolution. And when you filter at lower resolution, you can actually see an L1 stalk region, which is disordered upon release of the TNA. And here TNA is present. So the ligand is really inducing that. And this place is where the ligand is bound, competes out the TNA and doesn't allow it to rebind. that is actually the first case for the human ribosome, at least, to be able to analyze drug interactions now. And so in collaboration with a group from Jean-François Perron in Nice, we showed in a recent paper that actually the human ribosome is a very promising target for cancer research. And from there now we can start a lot of studies. So two aspects from that part, promising cancer target, and also we could start and envision structure-based drug design. So now we have worked extensively, and this is from here on, it's all unpublished work, on trying to push resolution. So we collected more data. We used more frames than in the beginning because the initial structure was actually a test data set with only 3 frames in the movies. It was much easier to process also at the time. We did much more sorting into 3D structures, and we did focus refinement. And with that, and I'll try to mention a few things here. So this aspect of local analysis is something we introduced actually a long time ago. To do classifications in local areas. And it's basically a focused classification approach, which then was extended in the field to do focused refinements. And this is really working great. What we have done here is to extend it to different regions. So first you can do the 2 subunits of the ribosome, and then you can go into subregions. And this is all done in Reliant in this case. So you see how much you can push resolution compared if you refine the entire structure as one single unit. And so here you can actually see very nicely what's happening. This region is being refined. The other parts are getting blurry if you try to refine that. And this type of focus refinement works particularly well if you do a good 3D classification beforehand. So if I just come back, the 3D classification here and into the sorting into pre- and post-translocation states is crucial to be able to do focus refinement properly. Okay, so that's the type of resolution one can get right now. So 2.5 angstrom local resolution. Here you see some typical map where you see the side chains in many places. And if you look into details, then you start seeing things like this. Lysine modification, methyl group. Here an arginine double conformation, as Sriram was also alluding to. This is things you can start seeing now by electron microscopy, not only by crystallography, as we did and still do actually in parallel. So yeah, that's clear features that are very interesting. Also on the 40S, which is a more flexible part of the structure, it's always very tricky to get any good features, structural features there. And now with the focus refinement, we see equally well. This is 3.1 angstrom average structure of the 40S head. So you see all the side chains and everything you wish to see in the 40S. Then there's a major aspect in terms of structural refinement of the atomic model. Now here you can see, for example, how the previous atomic model would lay. This is our own model and also quite similar to the one from a porcine ribosome. Any previous structure basically missed some of the errors in terms of registers. And now this is corrected here throughout the structure. So here you see that you have the right G and C, while here you had a register shift. So things are shifted sometimes by just one nucleotide, and the other strand then moves accordingly in the other direction. That is true for many regions. Then we also found an interesting region here of something that had been annotated as an RNA, which in fact is the ribosome part of a protein ER29, which fits very nicely and which is traced ab initio for that part. And it's an interesting region because this is actually holding together expansion segments at the periphery of the ribosome 60S subunit. So you can see all the interactions of that particular part of the protein which holds these pieces together. Yeah, and then a key aspect in this analysis was that for the first time we could actually see modifications, chemical modifications. So only methyl groups you can see here. So this is the 2' OH methyl groups are seen in various places. And they have a clear function. So I'll show a couple of examples. Here you have this triad of modified nucleotides. And you see they do all van der Waals contacts with the neighbor. Here to a cytosine. Here they pack between ribose backbones. and so on. So this is stabilizing the loop region. I'll come to that later. This is like an example of where you have modifications here on guanines, for example, which will be bridging other features in the structure. So in this case, phosphate backbones. So they stabilize loop interactions. So this is an acetyl group here that is interacting, hydrogen bonding. You have lots of other cases. For example, here, P interactions on a cytosine coming from a methyl group here in an M5 on a cytosine. Or here it's interacting and doing P stacking with a tyrosine. So that globally gives a lot of stabilization, and also these regions in particular, now for example that one, are directly involved in translation fidelity. So there are lots of functional data that you can actually explain with that now. They are related with modifications. All right, and then at the inter-subunit interface, you see, for example, here this is a 60S protein, EL41, which interacts with a modified residue from the 40S subunit. And then here you see the modification on that residue. And a different place, and also on the 40S, is modified in the same manner. And it's actually interesting to see that both sites are being modified by the same NAT10 enzyme during biogenesis of the ribosome. And both places were actually unpredicted in any of the databases, but they turn out to be universally conserved in this context. Right. And then another example, in the decoding center on the 40S subunit, you have this very beautiful pair of adenines being modified. Here you see dimethyl groups, and these are actually conserved positions in terms of evolution, and this in the decoding site, so crucial for decoding events. Yeah, and in a little example about pseudouridines, there are plenty of these, but it's to give you a feeling of what can be seen here. This is a normal uridine, and this is pseudouridine, so that, of course, will change the hydrogen bond patterns that you can have in a structure, in which case, for example, here, this residue will be able to interact with that pseudouridine. That's a 60S case, and here it's on a 40S. There's lots of examples like that. Now, there are plenty of such modifications, but which ones are really crucial, and how to analyze that further in terms of drug design? And actually, this is the next step afterwards. So, crucial in terms of interactions, we actually see that they happen very often in loop regions of RNA at the end of protein of RNA helices in very specific places in ketones and so on. So what you can see here, for example, in the center, you have a non-canonical base pairing, GU interaction with this modified G here. And the neighbor is also a non-canonical base pair, but it's tilted. And that tilt actually propagates afterwards, and it poses a tilt to a canonical base pair over there, because these ones are already pre-tilted. Yeah? So you have this propagation of a structure, and that means even this very conserved interaction actually becomes tilted, so it's modifying the way it interacts. Here you have a zoom on that particular area where you can see that. So there are two nucleotides involved here. All right, and then another particular aspect, and this starts to move towards medical implications, are these modifications on O6 positions of guanines or on the O4 position of uridines. These are very uncommon, at least to our knowledge, on RNAs. Maybe someone can comment on that later. But we found actually that they're known very well for DNA and to be related with cancer, obviously. So it's a question actually why we do see them in the structure, because ribosomes are purified from HeLa cells. So maybe that is a particular reason to actually see these modifications. They're not predicted in databases. Huh? Okay, and then other aspect in terms of drug design. We looked at binding sites, and so this complex actually contains 3 antibiotics simultaneously. So these are on different sites, on the 60S here, on the 40S, and another one on the 60S, which we spoke about before with the TNA binding site in here. Yeah? And so you can see actually some of the modifications are interacting directly with the ligand. Some are in the second shell, but reachable if you would consider to do drug design. And so basically they are in a druggable distance, I would say. Yeah? And they should be considered for drug design. So there are 3 places like this, and you see how well the ligands are actually defined. These are slightly substoichiometric, so you need to lower the contour level a little bit, not much, but a little bit, to be able to see that, and they're very well defined here. All right, so in terms of all the modifications that we have gone through right now, this is only on the 60S subunit. You see all the ones, and here we started classifying all the modifications that we can find on RNA. There are some conserved sites, very few. It's the pink ones. These are also present in bacterial ribosomes. It's only very few. Because the ones that exist actually are not all conserved. So it's only a few that propagate throughout evolution. We'll come to that in a second. And then you have predicted and found sites. So these are a lot of 2' methyl groups. And then a few unpredicted sites in terms of these 2' methyl groups, the blue ones, and a lot of new sites that were not predicted from any database. So there's actually 2 aspects there. Prediction is a good thing to have some databases to be able to tell whether you expect something, so you can check it. But it's not going to be enough to have a normal structure, I mean, without seeing modifications and just thread those predicted sites, because it will not give you the environment in the structure. So it's crucial to have that information right now. And in addition, there are a lot of new sites. So these unpredicted sites and also these keto-modified sites that I mentioned already. The same is done here on the 40S subunit. There are fewer on that part of the ribosome. And so now if you compare bacterial ribosomes and the human ribosome, so this is the E. coli ribosome. There were actually 2 studies on that recently, Termis and E. coli structures. There are about 35 modifications in the large subunit. While now in the human ribosome, we have spotted 116 that we see in the overall structure. And so you can see that here they're very much in the central part of the functional sites, decoding sites and things like that, and they start spreading. Yeah? So in the human structure, you see how far they reach out here. So basically, we suggest that there's an extension of modifications throughout evolution from the key catalytical centers towards the entire structure. So with that, I'd like to summarize a few of these aspects that we have seen in the structure. For now, to our knowledge, it's the highest resolution structure of the human ribosome and also the most complete atomic model. Then we have modified we have seen more than 110 modifications with many unpredicted new sites, mostly located in bulges, in interior and hairpin loops at the end of RNA helices very often. And in base triplets, some of these are described, you know, the ones with the tilting of the nucleotide bases. They are typically happening at base triplets. Then A minor motifs and kink turns. So it's typically structures which need to be stabilized. And that's certainly also the real functional and structural role behind that of the maturated ribosome, because now you look at the product of all the biogenesis and modifications. They act either locally, which is crucial for ligand interactions, and altogether collectively probably to fine-tune translation in eukaryotic systems. So the classification we have done is, as I mentioned, universally conserved sites, human or eukaryotic specific modifications, which are most of these, and new unpredicted unique sites. And for some of them, we cannot really identify which is a chemical nature. If it's a propyl group or if it's an acetyl group, very often it's clear from the environment in terms of hydrogen bonding, but some of these cannot be addressed right now. But still, it gives a full picture which can be analyzed in the future. Then there's clearly an evolutionary extended shell of RNA modifications in eukaryotes, which we can't see for the first time. And the localization of modification in ligand binding sites should really be considered for drug design. And there's now, this is more an outlook question. So why are they actually introduced? And what's happening during biogenesis? So they could be introduced there and they're just left over. You don't know why, so they were needed basically for the folding, for the assembly and maturation. But they could also have real roles once it's assembled. And it really looks like it's a second option that is the right one in terms of seeing the final function and probably also the dysfunction. Because here starts the business of trying to understand the medical implications, especially with these keto calculations. All right, and yeah, so this is basically our most recent work on the human ribosome. No time to discuss all other aspects we've been working on, like on transcription complexes, on polyribosome assemblies. We also worked on fluorescence microscopy, and especially developments for software to and develop interfaces to actually be able to process super resolution data with D-storm data, for example. There's a whole series of papers on that from Andronov in my team. And also feasibility studies in FIP, SEM imaging, and this is actually done by Igor Onov also, with a size instrument, which we had in a partnership for some time, and which it's installed right now on the platform. So in this context of integration and rEM, and this the symposium clearly shows that also, it has a key role for integration across scales. And as was mentioned, it's largely also due to high sensitivity cameras, which here gives this kink in the curve when you look at structures being deposited in the protein database. And also the contribution of high resolution structures, which really here comes since 2013, basically. So that is the type of integration we try to do at the Center for Integrative Biology with various teams in our department working on transcription, translation, and RNA with various biomedical targets associated. And it hosts the national and European infrastructures, Frisbee and Instruct, which I introduced in the beginning. So if you want to submit any proposal, which is going to be selected based on a project proposal, you can either submit it through Instruct or through the Frisbee, the French infrastructure. And just a word on the National Center for Biomedical Cryo-EM in Strasbourg. So it's also designed, as was mentioned, for what is being developed at the SRF right now, 24/7 access. On-the-fly image processing is being done also, and there are lots of links with Diamond, for example. Remote control, CS and faceplates, GIF, image processing tools, FIP-SEM, high-resolution electron tomography, fluorescence microscopy. And this is the latest things we have done with a postdoc in my group, and together with Rado, who came and helped us a lot to set up the faceplate on the microscope, but also to run it at all, because there were lots of issues with software to do automatic data collections. And here you see the faceplate part here next to the apertures, which are inserted here in the microscope, that place. In focus, Volta faceplate, defocus, and classical data collection. So the resolution of this one is actually the best one, and turns out to be much faster for the processing, and also more precise. 3D classifications go faster. I mean, it's a very interesting tool. Also for large complexes, so it's not only for small ones. So here you see a current map of one of the structures. And so I'll finish acknowledging Rado, who really helped us a lot to make it happen, to have the microscope run again after a long upgrade phase with basically K2, GIF, and the faceplate. And then the people involved in terms of image processing. So Leonid is involved in all the fluorescence microscopy, Brice in database analysis. Ottilie worked on the faceplate data that I just showed at the end. Jean-François handles the microscope. Karim works on nucleosome complexes, which I had no time to speak about. Alexander Myasnikov did all the processing of the high-resolution map together with Hanna Kratzat, who was an M2 student and did a lot of work on that. Gunnar did all the atomic model building of the human ribosome. Igor is involved in various aspects of transcription complexes in FIP, in developments of software, and Sasha Otsumtsev does a lot of crystallography software developments. So with that, I'd like to thank everyone and take the questions.
Thank you very much, Bruno. There is time for 2 to 3 questions. One over there.
So the 3 structures you have done is the faceplate. In terms of number of particles you use.
Yeah, 52,000 particles for each. We did this on purpose. We did the same number of particles for the processing. We collected more. So if you really push it, then you get like the 3.4 angstrom. Those ones are at 3.8 with the same number of particles, the best one. With defocus, 0.5 micron defocus.
So the increased contrast does not help you in reducing the amount of data?
No, it does. You get a better resolution with fewer particles.
How much? How much? I mean, if you want to push to the same resolution, say 3.8, 3.5.
Yeah, so it's always difficult to quantify such things. So these things are at 4.2, and this one, or 4.5 actually, and this is 3.8, 3.5 depending on the regions. But the key thing is the quantification of the time it takes also to calculate. So it's much, much faster. The only difference between those 2 data sets in terms of speed is that here you need to do CTF correction, and that is becoming basically the rate limiting step in this process. No, not yet. No.
It's on.
Difference, and then this confers to the ribosome's different properties.
Yes.
Do you see evidences of those differences in modifications, or you can't?
Yeah, it's a very good point. We really reckon that this is happening because, I mean, to really show it, we would need to analyze a ribosome from different cell types, ideally. But these keto modifications, we have not, I mean, they're not predicted and we didn't expect them, and it looked like that they're specific to the context of HeLa cells, so highly proliferative cells. And indeed, we think that, because your point about heterogeneity also, very often people think it's a matter of presence of a protein or something like this. Here it's much more fine-tuned. It's the presence of RNA modification that makes a difference. So that's leading to the next step. Yeah.